So now with glycogen out of the way, we can move on to gluconeogenesis. We've talked about how glucose is stored when there's excess levels of it in the blood, and we've talked about how glucose is released from glycogen stores when blood glucose levels are low. The question now becomes, what if there's no glycogen around at all? What if cells are depleted for ATP, cells are also depleted for glucose, and there are no stores, either in the liver or in the muscle, to replenish glucose levels? What then? And luckily for us, our cells have the ability to make glucose pretty much from scratch. That's called gluconeogenesis, and that's what we'll talk about in this chunk. The last chunk will be the regulation of carbohydrate metabolism from the whole body approach, and how our body as a multi-organ system deals with glucose levels and carbohydrate levels in general. But first, gluconeogenesis. Essentially, gluconeogenesis can start with pyruvate and from it make glucose. That sounds a lot like glycogen in reverse, but not every reaction is the reversal of glycolysis. Many reactions are, and that will help us in terms of studying, but we can't necessarily go back up the way we came down. And we're familiar with these ideas before. If the top of the mountain is glucose and the bottom of the mountain is pyruvate, it's one thing to come down that mountain and convert glucose into pyruvate. It's a whole nother thing to go back up that mountain, and you're certainly not going back up the same way. So gluconeogenesis is the reverse of glycolysis in principle, but it's not the reverse of glycolysis in terms of pathway. It's a different way to get back to the beginning of glycolysis. In other words, a different way to get from pyruvate to glucose. So again, luckily for us, there are 7 steps of glycolysis that are directly reversible. They're just going to go the opposite way from what we've learned before. And there are 3 steps of glycolysis that are irreversible, that can't go backwards. And this stops glycolysis itself as a pathway from flowing backwards, kind of like the valve in a heart that stops blood from backflowing. These are critical steps in glycolysis that stops glycolysis from backflowing. And based on that, some of you might already have some idea of which steps of glycolysis are not reversible. Step 10, Step 3, and Step 1. Those should look familiar, because those are also the control points of glycolysis. Those are the 3 regulated steps of glycolysis that allow us to turn glycolysis up or down. So in glycolysis, Step 10 is the conversion of phosphoenol pyruvate into pyruvate. Step 3 is the production of fructose 1,6-bisphosphate from fructose 6-phosphate. And of course, Step 1 is the phosphorylation of glucose to glucose 6-phosphate. Those 3 steps cannot go in reverse. Gluconeogenesis, all it really does is reverse those 3 steps. Since the other 7 steps can flow backwards, all you need to go from pyruvate to glucose is 3 new reactions that can reverse the actions of Step 1, 3, and 10 of glycolysis. But again, it's important to note that the remaining 7 reactions we're not really talking about here occur. It's just that they occur exactly as we've learned them already for glycolysis, going backwards. And they're catalyzed by the same enzymes. So getting pyruvate to back up and reverse Step 10, getting pyruvate to become phosphoenol pyruvate, is actually a 2-step process when we go in that direction. Going from PEP to pyruvate takes 1 step. Going from pyruvate to PEP takes 2 steps. In that first step, pyruvate and carbon dioxide actually come together. So we add a carbon, and we create a 4-carbon molecule called oxaloacetate. Oxaloacetate. We know we've seen that before. That's the resident intermediate of the citric acid cycle. Actually, we talked about all this before. Remember, when we were talking about replenishing the citric acid cycle, if we were to much off too many intermediates for anabolism, we talked about this very reaction and how we can convert pyruvate into oxaloacetate. I even showed you this slide and said we will come back to this material in a few lectures. Well, here we are, talking about it in the context of gluconeogenesis. So here's the reaction as it is laid out here. Pyruvate plus that carbon dioxide, a water molecule, and some energy allow oxaloacetate to be made, depleted ADP, and 2 protons. That essentially is the reaction that occurred. This reaction does require energy in the form of ATP, and it is catalyzed by an enzyme we've already named. It's catalyzed by pyruvate carboxylase. We named this enzyme when we talked about the citric acid cycle and the replenishment of oxaloacetate. But remember, we're trying to get back to PEP, so we got to do a second step. Well, oxaloacetate can be converted into phosphoenol pyruvate by using an enzyme called phosphoenol pyruvate carboxykinase. If you think of that enzyme name, it's telling you that two different reaction types are occurring, a carboxylation and a kinase reaction. Well, the carboxylation is making reference to that carbon dioxide we added a moment ago. In fact, we're going to remove it, and the kinase is going to take its place because we are, of course, trying to make phosphoenol pyruvate. This reaction also requires energy. In this case, the energy is coming from GTP. And so oxaloacetate, with its extra carbon dioxide there near the bottom, and some energy in the form of GTP, can remove that carbon dioxide molecule, add a phosphate group to the molecule as well, release the carbon dioxide as waste, and we have GDP as our expended energy source. So essentially, we can summarize these two steps of going from pyruvate to PEP as carboxylating and then decarboxylating pyruvate, leaving it as a phosphorylated and rearranged version of itself, PEP. The conversion of pyruvate to PEP is pretty close to equilibrium. It's very balanced, truly balanced. Very little energy is consumed by this reaction, very little energy is released. From a universe perspective, it really could care less about reactions with delta G's near zero. And so what this means is that the only real thing that influences the rate of this reaction is the concentration of the reactants and the concentration of the products. If we have more reactants, we're going to push the reaction towards product. If we have more products, we're going to push the reaction towards reactants. In this case, we can think of the reactants as PEP and the products as pyruvate. So if the cells have higher levels of PEP than pyruvate, we're going to drive the reaction in the glycolysis direction. We're going to convert PEP into pyruvate. If we have higher levels of pyruvate than PEP, we're going to push this reaction in the gluconeogenesis direction and convert that pyruvate into PEP. This is referred to as the law of mass action in general chemistry, and all of you should have been exposed to this general idea well before you came into this class. The law of mass action is a fundamental principle of chemistry in general, and so it is something that we definitely want to be familiar with in general and understand and appreciate that the law of mass action is influencing this reversal of step 10 in glycolysis. So, step 10 is reversed. We've made PEP from pyruvate. Remember, the other steps can reverse on their own. So step 9 can be reversed, step 8 can be reversed, we go all the way up, and then we hit step 3. Step 3 and step 1 of glycolysis are irreversible, so the question becomes, well, how are they reversed? Luckily, if you think about steps 1 and 3 of glycolysis, they're just phosphorylation reactions. Step 1 is the phosphorylation of glucose to glucose 6-phosphate, and step 3 is the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. So if step 1 and step 3 are simply phosphorylation reactions, to reverse them, we just need dephosphorylation reactions. The only problem is dephosphorylation reactions take a different class of enzymes, enzymes that we call phosphatases. So if kinases add phosphate groups, phosphatases remove those phosphate groups, and so it's phosphatases that we need to reverse these last two steps. To reverse step 3, fructose 1,6-bisphosphate is hydrolyzed to produce fructose 6-phosphate and the phosphate ion released as a result, and this reaction is catalyzed by the enzyme fructose 1,6-bisphosphatase. Gotta love that name. Really not much to remember there. A phosphatase that is working on fructose 1,6-bisphosphate, all in the name. This enzyme is inhibited by AMP levels and activated by ATP. Pause the lecture for a moment and ask yourself, does that make any sense? Think about that regulation scheme. Does that make sense? Well, it makes a whole lot of sense if you think about it. If ATP levels are high, you should not be doing glycolysis. In fact, if ATP levels are high, perhaps you want to take whatever residual pyruvate your cell did make and reverse it back to glucose so it can be stored as glycogen. So if ATP levels are high, take that pyruvate, turn it into glucose, turn that glucose into glycogen and store it for later. In other words, if ATP levels are high, turn on fructose 1,6-bisphosphatase so that glycolysis can indeed go in reverse. AMP levels. If AMP levels are high, that's a really bad signal. High AMP levels mean that you have not only burned through all of your ATP, you've gone back and chewed on it again, and you've burned through all your ADP as well. All your adenosines are running around as monophosphates. That's bad news. You need ATP, and you need it quick. And so in this case, high levels of AMP trigger glycolysis and block gluconeogenesis, making sure that all available glucose is converted into pyruvate. The reversal of step 1 involves hydrolyzing glucose 6-phosphate to free glucose. This is what occurs in muscle and in liver cells, when that glucose needs to leave into the bloodstream. This is catalyzed by glucose 6-phosphatase, again a wonderful name for this enzyme, a phosphatase that removes a phosphate from glucose 6-phosphate. Remember, when we phosphorylated these sugars, we needed to use ATP because these reactions were endergonic. We needed to couple them to ATP hydrolysis. And here, going in reverse is energy releasing. So as we reverse step 3 and step 1, the energy we put into these molecules is released. All of that energy that was needed to make those bonds in step 1 and 3 are released when we break those bonds. And so here's the overall process of gluconeogenesis kind of depicted if you go from bottom to top. Here's pyruvate. Pyruvate is converted to oxaloacetate. Oxaloacetate is converted to PEP, which becomes 2PG by phosphoglycerate mutase, by enolase, which becomes 3PG by phosphoglycerate kinase, which then becomes 1,3-BPG, all the way to glyceraldehyde, and then DHAP is isomerized from one of those glyceraldehydes. And then we reverse that to fructose 1,6-bisphosphate. And then we have fructose 6-phosphate through the action of fructose 1,6-bisphosphatase. We can make glucose 6-phosphate through an isomerization, and we can remove that phosphate group through the action of glucose 6-phosphatase. So gluconeogenesis is here starting with pyruvate and going up. Glycolysis we already know, but to summarize it in total, we start with glucose and we work our way down to pyruvate. 2 pyruvates made for every molecule of glucose consumed. So the reversal of step 10, step 3, and step 1 allows us to fully reverse all of glycolysis and fully synthesize glucose from pyruvate. 2 pyruvates needed in order to make 1 glucose, of course. This completely undoes glycolysis and gets us right back to the glucose we started with. We can store that glucose as glycogen, as we talked about in the previous chunk, and live on it another day. So if energy in the cell becomes automatically or all of a sudden at a surplus, and we don't need the pyruvate we just made, we don't need more ATP from that pyruvate, this gives us a mechanism to go backwards and take that pyruvate, convert it back into glucose, and store that glucose for later. So to summarize this lecture chunk, making glucose from pyruvate is the pathway gluconeogenesis. This is not a direct reversal of glycolysis. We need 3 individual steps to proceed backwards differently, and those 3 steps are steps 1, 3, and 10 of glycolysis. We talked about step 10 in detail. Do remember that steps 9 through 4 must occur in reverse, and then step 3 and step 1 can be reversed by the actions of phosphatases, giving us our glucose that can be stored as glycogen.